All right, engineers, in this video we're going to talk about cholesterol metabolism. So cholesterol metabolism is extremely important because cholesterol is the basic unit for a lot of different things. One of the things that we'll talk about is for bile salts, which are really important for being able to help with the emulsification of large fat globules that we take in through ingestion. They're also important for a lot of the steroid hormones that we utilize on our body, like testosterone, progesterone, estrogen, aldosterone, cortisol. And they're also important because they are actually packaged. There's specific molecules, specifically called lipoproteins, and we'll talk about a couple of them in this video. We'll have another video in more detail on that. But we'll talk a little bit about how they're involved in the actual transport of cholesterol. And then also it's a very integral component of the membrane. But we'll talk about all that. But the significance of this whole video is to see how we're going to be able to synthesize cholesterol with basic unit of acetyl-CoA and how that can be used for synthesis mechanisms. And we'll talk about a couple enzymes along the way. Okay, so cholesterol metabolism is primarily going to be occurring inside of the liver. We can take cholesterol in through an exogenous pathway through the ingestion of foods, and we can take them and transport them to different tissues via what's called chylomicrons. But the endogenous pathway of cholesterol is actually going to be taking place within the liver. So let's see how we do that. Well, if you guys remember from a lot of these videos, we could take a molecule called glucose, and we could take glucose. And what could we do with them? If you guys remember, we could take glucose and we could eventually convert that glucose into pyruvate through glycolysis, right? And then another thing we could do is we could take that pyruvate and eventually convert it into acetyl-CoA. But what can happen with the acetyl-CoA, if you guys remember? Acetyl-CoA can actually go through a specific sequence of cyclic steps, right? Which is called the Krebs Cycle. And then from the Krebs Cycle, what do you generate? You generate a lot of NADHs. You generate a lot of FADH2s. You even generate a little bit of ATP through substrate phosphorylation. But these guys are generally taking their information to the what? The electron transport chain. And what happens at the electron transport chain? You make ATP. So that was the overall goal of this process, right? Now, what happens though whenever we have too much acetyl-CoA? What happens? If you guys remember the main anabolic hormone, I want you guys to really remember this one. That hormone was called insulin. Insulin is the main anabolic hormone of the body, okay? So insulin is extremely important in this process of cholesterol metabolism. So what is he going to do? We'll talk about that in a second. But now let's see how if there's too much acetyl-CoA, we already know that it can go into two different pathways that we talked about. One was a tiny little pathway we can say over here that we can make ketone bodies, if you guys remember, through ketogenesis. And then what can happen with these ketone bodies? If you remember, they can be taken up by two different specific tissues. The brain, the central nervous system tissue, as well as the muscles. And can be utilized to make what? They can be utilized to make ATP. But there's another side pathway that we can push acetyl-CoAs into, and that is into cholesterol synthesis. Now let's talk about that. So let's say that I take this acetyl-CoA, I put it right, well actually no, let's bring it over here then. Let's say that I take these acetyl-CoAs and we start here and see how this is all happening. So now acetyl-CoA, let's say I take an acetyl-CoA here, and I take another acetyl-CoA. And I'm going to react these two molecules together. So I'm going to take these two molecules and I'm going to react them together. So let's say I react these two together and I'm going to fuse them together. When I fuse acetyl-CoA and acetyl-CoA together, there's a special enzyme. There's a lot of different names for it, but I like the simple names. It's easier for me to remember, so I like thiolase. It's just an easier enzyme to remember as compared to like acetyl-CoA acetyl transferase. I just like the thiolase. Okay, but there is another enzyme that you could utilize for this step. Okay. Now, in this step, what am I doing? I'm taking acetyl-CoA and reacting them together in this first step of the reaction. But what am I going to get out of this? Out of this, I'm going to produce a molecule called acetyl-CoA. Now this might seem very familiar if you guys have already watched our ketone body videos. The actual beginning steps of this process is going to be almost exactly the same as the ketone bodies. Now we're not going to go through every single step to make cholesterol. It's about 30 steps long. So to do that would be ridiculous. We want to get the significant components out of this video. But here's what we would need to notice. Look at this. We have acetyl-CoA and acetyl-CoA. How many acetyl-CoAs is that? How many CoAs is that? That's 2 CoAs. Then what I did is I converted that into acetyl-CoA. There's only 1 CoA there. So what did I lose in this step? I lost a CoA. So in this step, I lost a coenzyme A. And I made acetyl-CoA. Now, in the next step, I'm going to do this reaction. And what I'm going to do is I'm going to combine another molecule. And this next molecule is exactly the same as what we had over here. Guess what I'm going to have coming over here? I'm going to have acetyl-CoA. So I'm going to feed another acetyl-CoA into this reaction. So now I'm going to take this acetyl-CoA and feed it into this reaction here. Okay, so let's actually show this in the same color then, so that we make it look all nice and purty. Acetyl-CoA here with the acetyl-CoA is going to react. And what am I going to do? I'm going to lose another CoA. So out of this reaction, what's going to come popping out? I'm going to pop out a CoA. Now I'm going to form a very special molecule. Now there's actually 2 names for this, the abbreviation. I'm just going to call it HMG-CoA. But there is another name for it. I will mention it, but it's easier to do this one. But it actually goes 3-hydroxy 3-methyl-gluteryl-CoA, or beta-hydroxy beta-methyl-gluteryl-CoA. Now you guys probably get why I would just call it HMG-CoA. It's easier to say that. But the whole point is that we do have a hydroxy group on the 3rd carbon, and then we have a methyl group on the 3rd carbon. And then it's a gluteryl-CoA. Okay, what enzyme is catalyzing this step? A very, very important enzyme. And this is called HMG-CoA synthase. Okay, he is catalyzing this step. He's stimulating this step to fuse acetyl-CoA with acetyl-CoA and make HMG-CoA. Now we go into the most important, one of the most regulated steps of all metabolism in the entire body. Okay, let's make sure that we understand that. So I'm going to do this in a really, really thick red arrow here. This is one of the most regulated enzymatic steps in the entire body. Okay, the enzyme that's regulating this step here is specifically called HMG-CoA reductase. So HMG-CoA reductase is the rate limiting step here. It's very, very important. So I'm going to put below it, it is the rate limiting step. So it's one of the slowest steps, but it's one of the most important and most regulated steps. So this is one of the most regulated steps in our body here, this step right here. Why? Because it's very, very crucial that we regulate how much cholesterol is being made. So how do we do that? Well, you know, there's actually drugs. You know, when people have really, really high cholesterol, they have to take certain types of drugs to be able to lower the cholesterol. You know how they actually do it? One of the drugs that you guys have probably heard of called Lipitor. So you've probably heard of the drug called Lipitor. You know, like its brand name, the Lipitor. Lipitor is actually what's called a statin. So there's different types of statins. And statins are basically just drugs that can come and inhibit this enzyme. So this HMG-CoA reductase can be inhibited by certain types of drugs, which can inhibit the cholesterol synthesis, which is good for people who have high cholesterol levels. They've been eating too much Burger King, too many Whoppers, right? So they need to cut that down, take their Lipitor and watch what they're eating. But anyway, this HMG-CoA reductase is very, very important because it's what's controlling the conversion of HMG-CoA into another very important molecule, which is going to be the precursor. So this molecule is called mevalonate, or mevalonic acid. It doesn't matter. I'm just going to call it mevalonate. So this next enzyme, again, is called mevalonate. Now, mevalonate is actually important because he's going to be the precursor for the synthesis of cholesterol. Now, like I said, we're not going to go over every single enzymatic step. It's ridiculous. There's so many of them. We'll cover some of the more significant ones. Like, you know, mevalonate can actually be converted into another molecule, which is called isopentyl pyrophosphate. So that molecule is called isopentyl pyrophosphate. Now, why am I mentioning this? Because whenever this mevalonate, he goes through a couple steps and it eventually gets converted into isopentyl pyrophosphate. Isopentyl pyrophosphate is going to be kind of the building block for this also, because what happens is I'm going to take a whole bunch of different types of isomerased forms of the isopentyl pyrophosphate or just the isopentyl pyrophosphate itself, and I'm going to combine a ton of them. I'm talking boatloads of these isopentyl pyrophosphates, and eventually I'm going to form a very special molecule. And this process right here, I'm completely bypassing a lot of important information here, or I'm sorry, not a lot of information, because, you know, just for me to convert this isopentyl pyrophosphate into this next molecule is called squalene. So isopentyl pyrophosphate, technically in between here, if I were to actually kind of have an in-between step here, isopentyl pyrophosphate, let's actually just do it here in this step. Let's just do it here. We say that we take the isopentyl pyrophosphate, and I actually convert that into these molecules which are called isoprene units. Isoprene units. And these isoprene units are important because it takes a ton of isoprene units to eventually get converted into a very important molecule called squalene. Okay, so now we'll talk about the squalene. So squalene, in order for me, so this is a complete oversimplification because simply going from these isoprene units to the squalene can take up to 21 steps. So let me write that down because it's significant. Me going just from the isoprene units to the squalene, it can take up to 21 steps. Now, one of the more important enzymes in throughout this process is called squalene synthase. And the squalene synthase is an enzyme that's helping within these processes to convert these isoprene units into squalene. Now, squalene is then going to be the precursor to make another molecule. And this molecule is called 7-dehydrocholesterol. So 7-dehydrocholesterol, you guys have probably heard of that whenever we talked about the parathyroid hormone with helping to make vitamin D. So you can actually make vitamin D from this point here. But look, we're going to make another molecule called 7-dehydrocholesterol. Then what happens? So that's 7-dehydrocholesterol. Okay, now, here's where we get into the good stuff. So now this 7-dehydrocholesterol, what can happen with this molecule? This 7-dehydrocholesterol, so I'm going to put this squalene synthase under here so that we have some room to come back down again. So this is called squalene synthase. Now here's where it's important. So let's make this a really big line here. 7-dehydrocholesterol can be converted into the molecule that we know as cholesterol. So in this process here, what am I doing? I'm taking the 7-dehydrocholesterol and I'm converting it into a molecule called cholesterol. Now, this cholesterol that I'm making, it's crazy to see how we just take these 2 carbon fragments, acetyl-CoAs, and we synthesize a huge 27 carbon molecule. You know what? You guys want to know what cholesterol looks like? I'll tell you. I know you guys are excited. You guys are chomping at the bits to see the structure of cholesterol. So look here, because I know you guys are chomping at the bit for it. So look, I'm going to have 3 6-carbon rings like this, all right? Then I'm going to have another 6-carbon ring like this, and then I'm going to have a 5-carbon ring like this. Then I like to put a methyl right here and a methyl right here. So now let's number all of these puppies here. This right here is number 1, 2, 3, 4. Then I'm going to come over here to 5, 6, 7, 8, 9. And I'm going to come to this last point here, 10. Okay? Then I'm going to come up over here, 11, 12, 13. I'm going to go over here, 14, 15, 16, 17. Then I'm going to come over here, 18, 19. But I told you, oh wait, Zach, you said it was 27. It is 27. Coming off of the 17th carbon, I'm going to have a branch like that. 1, 2, 3, 4, 5. So 1, 2, 3, 4, 5. And then like this. So now let's count these carbons over here. 17, 18, 19, this is going to be 20, 21, 22, 23, 24, 25, 26, 27. Why am I telling you all of this? Because there's important points throughout all of this. On the third carbon, there's a special thing that's going to determine the difference between certain types of steroid hormones. On that third carbon, there's usually a alcohol, okay? And then in between the fifth and the sixth carbon, there should be a double bond. So now, I like to just call this A ring, the B ring, C ring, and the D ring. And then you got this little antenna structure. This is our cholesterol molecule. Now, we synthesize this cholesterol. What is this cholesterol used for? Because it's extremely important. We said that it's important to be able to take this cholesterol and synthesize a bunch of different structures. So now let's take this cholesterol and see what we can actually do with this cholesterol. Okay. So I told you that I could take this cholesterol molecule. I'm not going to drill every single carbon again. I'm just going to take this carbon unit so that you can see that we have cholesterol here. And what we're going to do with this cholesterol is we're going to take this cholesterol and use it to make a whole bunch of different molecules. And again, we can, if you want to, A ring, B ring, C ring, D ring. Okay. What I can do with this cholesterol is I can put it into the actual cell membrane. You know the cell membranes are really important. Why? Why is the cell membrane important? Let's say that I have here the cell membrane. And in the cell membrane here, you know we have phospholipids that are making up the actual cell membrane. If I were to kind of zoom in on this a little bit, let's say here's my glycerol, okay? And then coming on one side I have the fatty acids, right? But you know that this, specifically the cell membrane, is a lipid bilayer. So on the other side I'll also have another group of phospholipids. But now, here's the important thing. Phospholipids are, you know, they're basically, they can make things not as fluid-like, but cholesterol is making it better and not being so fluid-like, making it a little bit more rigid. So if I take this cholesterol molecule and I put this cholesterol molecule into this structure, so let's say I erase this part here, and I put my cholesterol molecule in here. So now what I do is I take my cholesterol molecule and I incorporate this cholesterol molecule into this structure. What is that going to do? What this does is actually, first off, it does two things. Two things that this does, being incorporated into the membrane. One is it makes the membrane less fluid, like less fluidity. Okay, that's one thing. So it's going to make it less fluid. The other thing, it's going to prevent drastic changes in phase transitions. So changing from different phases. So it prevents phase transitions. You know what's another important thing? It's also combining with a molecule that is present in this area. Let's say here I have a molecule, a special molecule, that it's combining with to form a very special structure. What is this green molecule here called? This is called glycosphingolipids. So these glycosphingolipids are combining with the cholesterol, and as a structure, they're forming this whole thing here. If I were to kind of encase it here, they're forming what's called the lipid raft, which is important in certain types of signaling pathways as well as a lot of other processes. So again, what's one thing that we can do with this cholesterol right away? One thing I can do with this cholesterol is I can incorporate this cholesterol into the cell membranes, because it's naturally, in normal physiological levels, is helping the membrane become less fluid, helping it to be more rigid and hold up its actual structure. The other thing is preventing excessive phase transitions. Okay. Also, it's combining with these glycosphingolipids within the cell membrane to form lipid raft structures, which are important for a lot of different functions like cell signaling processes. What else can we do with the cholesterol? So that's one thing that we can do with it. Another thing that we can do with this is we can take and convert it into steroid hormones. Steroid hormones are very, very important. So I can actually make steroid hormones. What kind of steroid hormones could I make? You know, there's many, many different types of steroid hormones. We talked about some of them in endocrine. I can make testosterone. I can make estrogen. I can make corticosteroids, which includes your aldosterone and your cortisol. I can make tons of different things, progesterone, a lot of different molecules that can come from this, right? So we can make steroid hormones. We can make progesterone, testosterone, estrogen, a lot of different corticosteroids like cortisol, aldosterone, gonadotropins. So many different types of things that we can do with this. So that's another basic unit for the cholesterol. So cholesterol can be used for cell membrane structure, steroid hormones. What else could it be used for? It also could be used for bile salts. You know, there's these things called bile salts, and these bile salts are important because bile salts are important. You know, there's what's called cholic acid. There's called cholic acid. And then there's another one called deoxycholic acid. And these are bile salts, and they're important for what? You know they're important, we actually make this in the liver and they're incorporated into the bile, which is excreted into the small intestine, like specifically the duodenum. What does that help for? It helps with the emulsification of lipids. You know, taking lipids that are very, very water insoluble and trying to make them more water soluble by spreading them out into smaller fatty droplets that we'll talk about called micelles. Okay, and the last thing. We'll have another video specifically on this in detail, but you know what else we can do with that cholesterol? We can package this cholesterol and we can send it to other tissues. So I can actually package this cholesterol into these special lipoprotein molecules. So let's say I take that cholesterol, but you know, in order for me to take this cholesterol and put it into this actual vesicle, this lipoprotein structure, I need a special enzyme. And this enzyme is called ACAT. And ACAT is an enzyme that basically converts cholesterol into a cholesterol ester. So again, what am I converting? So if you guys remember here on the A, on that third carbon, I had an alcohol. What I'm going to do is I'm going to take in this vesicle here, I'm going to convert inside here, I'm going to take and I'm going to basically take and add an acyl group onto it. So you know ACAT is basically an acetyl-CoA, and specifically what I'm doing here is I'm transferring on a acyl group. So in other words, let me show you what I'm doing here. So I'm going to take that actual group here and I'm going to put on a double bond, like a CH3 there. So let's say I add on a 2-carbon group there. Now this is a cholesterol ester. And if I were to draw the rest of the structure here, you guys would get the point here that basically I'm packaging this molecule into a nice big vesicle. So let me draw a big vesicle here. Lipoprotein molecule. So now this lipoprotein is going to have my cholesterol ester in it. So again, what is this molecule here called? This is a cholesterol ester. And this ACAT enzyme is transferring that acyl group, that 2-carbon group, onto the cholesterol molecule. So it's acting as a acylcholesterol transferase enzyme. And then after we package this into a protein covering, so now this is actually going to be the actual cholesterol component, and around it I'm going to have my protein component. And on the protein component I can have specific types of apoproteins that we'll talk about depending upon the type of lipoprotein it is. So for example, this can be one type. You know what this one is actually? We can actually call this one Ldl Bad Cholesterol. Okay? And this one is actually very dangerous if there's really, really high LDL levels. That's why whenever people have high cholesterol, mainly LDL, you want to give them statins or Lipitor, because if you remember, that's going to inhibit the HMG-CoA reductase enzyme, so you don't make as much cholesterol. So you can't utilize it in the lipoprotein pathway. So again, what is this molecule here called? It's called a lipoproteins. So it's very important for making lipoproteins, which can go and get transported to other different tissues. You know there's another one that you can make here? There's another one, it's actually called Vldl. We talked about that in the triglyceride synthesis video. The only difference is that instead of having a significant amount of cholesterol, you also add in triglycerides. Triglycerides are also added into the VLDLs in significant amounts. There's a lot of triglycerides and not as much cholesterol esters. But nonetheless, your liver can make VLDLs and LDLs. It can also make other lipoproteins, which you guys have probably heard of, called HDLs, which are going to help with being able to pull some of the cholesterol off of the vessel walls. But we'll have an individual video on that. But again, understanding all of this is important, but understanding the significance of why the cholesterol metabolism is occurring is more important. Understanding that I can use this for synthesis pathways, incorporating into the cell membrane, making steroid hormones, making bile salts, and actually helping to make lipoproteins for other different tissues. Because if you imagine, LDL is actually taking and transporting some of that cholesterol to like the adrenal cortex. You know, in the adrenal cortex, they need that cholesterol. They need that cholesterol to be able to make their steroid hormones. So it's very important that we have that cholesterol. Also taking it to the testes to be able to make testosterone and estrogen. So without that, there could actually be potential like life-threatening problems. But too much of it is also pretty bad. And again, knowing that out of this is one of the most important metabolic steps in the entire body. You know, not only lipitor is actually inhibiting this. Oh, and another thing, I'm so sorry I didn't mention this. This step right here, we need NADPH to NADP positive. If you guys remember, this was a very, very strong reducing agent. And we need him in this step because of his reducing power. NADPH is utilized in a lot of different pathways. You know, not just in steroid synthesis, not just in triglyceride synthesis, but also for nucleotide synthesis as well, and even the synthesis of neurotransmitters too. So NADPH is actually utilized in this step. But remember, this is a highly regulated enzyme. Another thing that can actually inhibit this guy at this point here is also going to be cholesterol. Cholesterol itself can inhibit this HMG-CoA reductase. So high amounts of cholesterol can act on this HMG-CoA reductase and actually inhibit the synthesis of himself. He does it by activating specific proteolytic enzymes. So he activates proteolytic enzymes that will actually help to specifically inhibit this HMG-CoA reductase. But like I told you before, insulin is also extremely important because insulin is also going to try to stimulate this enzyme, activating this enzyme through certain types of second messenger intracellular pathways to stimulate this enzyme. Whereas the actual guy who tries to oppose insulin is glucagon. And you guys know that glucagon wants to be able to do what to this enzyme? He wants to inhibit this enzyme, inhibit the actual synthesis of cholesterol, use it for making glucose, use that acetyl-CoA to make glucose or to help to be able to generate ATP instead, primarily though to make glucose. Okay, so very, very highly regulated enzyme here. All right, so in this video we covered a lot of information. I hope it all made sense. I hope you guys really did enjoy it. I hope it clicked. If it did, and if you guys enjoyed it, hit that like button, subscribe, put a comment down in the comment section, guys. We really look forward to hearing from you guys. All right, Ninja Nerds, until next time.